Okay, thank you. So next up is Jeff Barrett. Jeff's director for the Center of Therapeutic Target Validation here at the Wellcome Trust Institute. And previously he was at the Whitehead, and before moving to Oxford and then coming to Sanger as a group leader. So thanks, Jeff.
Thanks, Jeff, and thanks very much for the invitation to talk about CDTV here. So what I'd like to do is just describe a little bit about what the Center for Therapeutic Target Validation is, and as I do that, hopefully illustrate how central partnerships between the three founding institutions are to all of the elements of what we're working on. So the problem that CDTV was put together to address, or at least to do some work on, is that drug development rarely succeeds, depending on which literature you look at. This is one review from last year. Something like 1 in 10, or maybe a bit better, of compounds that make it into phase 1 clinical trials make it through each subsequent phase to eventually be marketable drugs. And these can fail for a variety of reasons: safety, efficacy, commercial considerations, but a surprising fraction of them fail because they simply don't work. That the starting hypothesis that if you can develop a drug that safely modulates this target, it will help in sufferers of some disease, doesn't happen to be true. And the realization, I think, that started with scientists in GSK, and this is some data from a paper by Matt Nelson and other colleagues at GSK that just came out about a month ago, is that human genetics can provide a very strong starting point for some of these questions of validating the therapeutic efficacy of particular targets. And this is several different types of data, starting from OMIM, or Mendelian rare penetrant genetics, including GWAS, which has obviously become a very popular type of genetic analysis in the last 10 years or so, and is where a lot of my academic research is, and also various other types of genetic annotations that, in essence, if you have a priori genetic evidence of this gene being involved in this disease, it makes it much more likely, twofold or more, that that target will end up having a therapeutic benefit. And the goal, of course, is instead of looking at this retrospectively, these data are from looking at successful and failed drugs and seeing if they have any genetic evidence after the fact. Can we turn it around and do it prospectively? And so CDTV was formed about a year ago as a partnership between GSK, the European Bioinformatics Institute, and the Wellcome Trust Sanger Institute. The latter two are both here on the Genome campus. And we're not certainly the only ones to have had this insight that human genetics and the really rapid advance in the last 10 years or so offers an opportunity to try to make a big difference in this earliest phase of drug development. There are other public-private partnerships, both in Europe and the US. There are lots of individual pharma companies who have tried to make a new investment in genetics and using genetics as a starting point. And so what I want to do today is just talk about 3 things which I think are unique about CDTV and also really touch on this issue of partnerships. So the first is an idea of industrializing big data integration. So academic groups are producing huge amounts of genetic and genomic data, and really sometimes that's not well suited to a small team of PhD students and postdocs. And they're all driven by publications, and they write one paper and they move on to the next thing. And really, for a lot of these data sets, what we want is stable, well-integrated, up-to-date information that can be rolled out to as many users as possible. The second is this idea of human genetics in human cells, and this is a set of experimental projects that CDTV is starting. And we really want to focus, not to say that the model organism work isn't important at various stages of drug development, but we want to emphasize, because it's a strength at Sanger, using human cellular models and starting with human genetic data. And the final thing, which is perhaps most relevant today, is to really try to redefine some aspects of partnerships between pharma and academics. So Lon Carden, who's one of the people from the GSK side who really got this in motion, had a habit of saying he used to be an academic, and he would have relationships with pharma where, as he put it, they would throw money over a wall and say, do some good science and hopefully something will come out of it. And it really doesn't work well for either partner in that side. Rarely does any useful information come back to the funding companies, and in fact, it doesn't really get a good kind of anything, a good academic incentive for the academics who get the money to do a particular research project. So thinking about each of these, EBI has a number of the sort of key databases that exist for instance, GWAS, so common disease, common variation data sets, this thing called COSMIC, which is about cancer genetics, something called EXAC, which is about general variation in a very large set of 60,000 individuals to just sort of study for an individual gene, what's the kind of variation that's out there in the healthy population, things that are focused on rare diseases, so OMIM, ClinVar, things like that. And these databases have lots of information that's useful for thinking about new targets, but they rarely talk to each other, even though they're often sitting under the same place. And there's a ton of other data, not just the genetics, but things like gene expression profiles in different tissues, different information about protein structures. This database called Campbell, which is run at EBI, that is about existing drugs and their indications in trial data. And the issue is that even though a lot of these things are housed in the same place, they don't communicate to each other seamlessly. And one of the things about CDTV was to put the resources in place not just to deliver on any one of these pieces, but to fundamentally bring them all together so that we've essentially embedded staff in every one of a dozen teams at EBI to both build on their own resource and make it possible for those resources to be linked together. And we want to do that using both the sort of computational infrastructure that we can support in the long term, and also with statistical techniques to make sure that we're getting as much out of these data as possible. And at the moment, we then feed that into a web interface that scientists at, say, GSK, but actually one important part of CDTV is that all of our resources and all of our data will be made publicly available, and ultimately our computational tools will be available not just to GSK, but other companies, other academics. And this is really, I think GSK has an insight that this early phase of biology, they want to get the best cutting-edge academic work, and they are willing to take a step away from whether there's IP in that early target biology to make it as good as possible for them so they can focus on where they see their competitive advantage, which is in making drugs. So that's one aspect. Another I said was human genetics in cells. I like this particular example because I work on inflammatory bowel disease in my academic life. And there was a really exciting antisense oligo targeting a gene called SMAD7 that a trial was published just this year. And it was really stunningly successful. My gastroenterology colleagues, in fact, thought it was a bit too good to believe, so it'll be interesting to see subsequent trials. The thing that I think is interesting is it was done in Crohn's disease, which is one of the two common types of IBD. It turns out there's a lot they've been following this gene for a long time. There is evidence just recently that a strong genetic association between genetic variation near the target, SMAD7 and Crohn's, but actually it's also associated indeed a bit more strongly with the other common type of IBD, ulcerative colitis, and that's a potentially really quick, easy win for repositioning that they should be thinking about trials in UC as well as Crohn's. And that's the kind of thing that you can get by kind of mining the literature, but in fact, some of this stuff is buried deep in supplementary table 57 of a big GWAS paper, and getting that into the hands of people working in pharma companies right away is one of our main goals. We also want to take it not just with the genetics data, but with cellular models. This is one example of a collaboration sticking with IBD between us and the Addenbrooke's Hospital campus here in Cambridge. They take colonic biopsies from patients. There's a scientist here, Trevor Lawley, who's faculty at the Sanger Institute, who then creates these colonic organoids, which are three-dimensional cultures of epithelial cells, and you can do all kinds of interesting probes of those as a model, so you can put a microbiome sample or a pathogen or a particular stimulus to see its reaction in the context of the genetics we know is relevant to IBD. And then the last thing I want to mention is this idea of a new type of partnership, which I briefly alluded to. And it's fundamentally about not just having some cash for us to do a bit more science, but really about, as I already mentioned, GSK's belief that they are going to win by making this open investment, but also about the academics changing the way they think about working with industry. And fundamentally, if we don't work with people who make drugs, then the genetics research we do won't make an important difference in society. And for myself, working on this broad area of complex disease genetics for the last 10 years, it was clear that I needed a different way of working than just the traditional academic path. That has, of course, a ton of benefits to it, but to really ultimately make a difference. And this is just one example of the way we've designed all of our informatics tools is not by just us thinking, oh, what's an interesting thing to do, and we'll program it up and then just throw it out there and hope someone cares. But we've run a huge series of user experience and user interface workshops, had these deep dives where GSK scientists come and try to work on genetic problems in their targets of interest while we kind of spy on them, essentially, to figure out what are they doing, where do we think we can give them added value, and really try to build the system together. And I'll just mention that it's not just on the informatics side, but also a great example of a project where we're doing some biological experiments, but it's really involving, again, as I think Jeff alluded to, not just sort of, well, we're doing something that's like vaguely of interest to both parties, but that fundamentally we're sharing the work. GSK, like many other companies, has a wide variety of cell lines that they use for various assays. Some of these have been immortalized 50 years ago or something. And one question I like to ask is, in an epigenetic or transcriptomic way, how do these lines represent the primary tissues they're supposedly modeling? And so one thing we can do is generate these epigenetic profiles of key cell types. We did that by having GSK scientists come to Sanger, get some training in doing the chromatin immunoprecipitation and other aspects of preparing the RNA and DNA material, taking that back to a GSK lab where they have all the cell lines, they can extract the DNA and RNA, package up the products we need, send it back to Sanger for sequencing, and then we come together to do analysis where you produce these sort of genome-wide profiles of essentially which areas of the genome are switched on and off in this particular cell type. And that's an example where neither one of us could have done that completely on our own, and so the ability to share with each other is really a fundamentally important part of this project. And so I'll just finish by saying that on every one of these levels, I think partnerships and collaboration underpin what we're aiming to do. And I'll just emphasize that GSK, I think, saw an opportunity to get this moving and decided to invest on their own to start the CDTV 12 months ago. But their vision was always, and is mine, that we'll open this up and bring other, both academic and industry partners on board. And what we really want to do is show other groups and other companies that this is a profitable way of working and that there is a benefit from them to joining. So thanks very much.